I am Jocelyn Makasare, Assistant Professor, Parvatibai Chogle College of Arts and Science, Margao. Program Bachelor of Science, Subject Botany, Semester 4, Course Code BOC 104, Course Title Plant Physiology. Title of the unit Photosynthesis, Module name Photosynthetic Pigments. Outline Photosynthetic pigments, chlorophyll A, chlorophyll B, Xanthophylls and Carotene. Learning outcomes discusses about the photosynthetic pigments. There are three types of photosynthetic pigments. Firstly, we have chlorophylls, phycobilins and carotenoids. We shall see chlorophylls first. The characteristics of chlorophyll pigment. They are insoluble in water and soluble in organic solvents. They will absorb light in the blue region of the visible spectrum from 420 to 450 nanometers and in the red region of 650 to 700 nanometers. Chlorophyll A and chlorophyll B are the two main important photosynthetic pigments which absorb light. Chlorophyll A will convert the light energy into chemical energy. These are the different types of chlorophylls. Chlorophyll A, B, C, D and E, which are found in algae and higher plants. Then we have bacteriochlorophyll A, B, C, D, E and G, chlorobium chlorophyll and bacterioviridin. These three are found in bacteria. Chlorophyll is formed from protochlorophyll in light. We have protochlorophyll, which in the presence of light at around 211 nanometers forms chlorophyll. Structure of chlorophyll, it is chlorophyll A, chlorophyll B consists of a magnesium porphyrin head which is hydrophilic in nature. Now this porphyrin ring will consist of 4 pyrrole rings which are joined together by CH bridges or carbon-hydrogen bridges, and it also contains a long phytol chain which consists of carbon atoms. Now this phytol chain or the tail is lipophilic in nature and it is attached to the porphyrin ring at the 4th pyrrole ring. Now these are the structures of chlorophyll A and chlorophyll B. This is the magnesium porphyrin ring and this is the phytol chain. Now there is a difference between chlorophyll A and chlorophyll B, the structure. Now if you see here at the 2nd pyrrole ring, we have a CH3 or a methyl group attached, and at the same position in the 2nd pyrrole ring there is an aldehyde group or a CHO group. Now this is the main structural difference between chlorophyll A and chlorophyll B. The same is explained here. This is the formula for chlorophyll A and it contains a methyl group at the C3 in the 2nd pyrrole ring. And chlorophyll B, this is the chemical formula which contains the aldehyde group or the CHO group at the C3 in the 2nd pyrrole ring. The photosystem 2 consists majorly of chlorophyll B pigment. We now come to phycobilins. Phycobilins are soluble in water. Now when we study the structure of phycobilins, they resemble chlorophylls in the tetrapyrrole structure. They will have an open chain of 4 pyrrole rings but without magnesium and the phytol chain. Phycobilins will absorb light in the visible spectrum at 500 to 600 nanometers. Now these are the major photosynthetic pigments of the blue, green and red algae. Now it is these bile pigments which give the algae a distinctive color of blue and red. There are two types of phycobilins, phycocyanin and phycoerythrin. We have phycocyanin which imparts the blue color and phycoerythrin which imparts the red color. We now move on to carotenoids. There are two types of carotenoids, carotenes and xanthophylls. Xanthophylls are also known as carotenols. Carotenes are orange pigments which are found in photosystem 1, and xanthophylls or carotenols are yellow pigments which are found in photosystem 2. Characteristics of carotenoids. Carotenoids are insoluble in water. They are soluble in organic solvents. They are mostly found in algae and in higher plants, and they will absorb blue, blue-green and green light of the visible spectrum from 450 to 500 nanometers. The structure of carotenes. These are open chain conjugated double bond systems which end on both the sides with ionone rings. The general formula, molecular formula for carotenes is C40H56. Carotenes are stereoisomers. That means they are compounds which will differ in arrangement of their molecules in space. Structure of xanthophylls. Now, the structure of xanthophylls is quite similar to that of carotenes, but they will only differ in having two oxygen atoms in the form of hydroxyl, carbonyl or carboxyl groups attached to the ionone rings. The general formula of xanthophylls will be C40H56O2. What are the functions of carotenoids? Firstly, carotenoids absorb light and they will transfer this light to chlorophyll A. And second, they will protect the chlorophyll molecules from photooxidation. Now, how photooxidation can be prevented? Firstly, the carotenoids will quench the first excited triplet state of chlorophyll photosynthesizer. They can also quench the singlet oxygen directly, and then there are some carotenoids which will act as a substrate for oxidation by singlet oxygen species. Location of the photosynthetic pigments in the chloroplast. These photosynthetic pigments occur as protein pigment complexes in photosystem 1 and in photosystem 2. Now, they will be dispersed in the lipid bilayer of the thylakoid membranes of the grana in the chloroplast, and they can also be present in the stroma lamellae. Absorption and utilization of light energy by photosynthetic pigments. Now, the chief source of light energy for photosynthesis is the sun, but only a very small portion of the sunlight is absorbed by the pigments, and this light which is absorbed is from the visible part of the electromagnetic spectrum, that is from 400 to 700 nanometers, and therefore it is known as photosynthetically active radiation or PAR. Now, there are certain photosynthetic bacteria which use infrared light of shorter wavelengths. Out of the total sunlight or light energy received by the earth, only 1% of the total solar energy is absorbed by the pigments and utilized in photosynthesis. There is weak absorption of light energy by these pigments in the green spectrum. Hence, the chloroplasts appear green in color. What are accessory pigments? All the pigments except chlorophyll A are known as accessory pigments or antenna pigments. Now, what these accessory pigments will do is they will absorb all the light and they will transfer almost all of it to chlorophyll A molecules by resonance. It is also known as FRET or Foster resonance energy transfer. Almost all the light which is absorbed by the accessory pigments is transferred to chlorophyll A molecules by resonance. It is also known as Foster resonance energy transfer or FRET. Chlorophyll A molecules can absorb the light energy directly. In photosynthetic bacteria, the light is absorbed by bacteriochlorophylls and bacterioviridins, and other pigments are absent in bacteria. These are the references.